The structure of DNA. We know that DNA is the information molecule. It stores instructions for making other large molecules called proteins. DNA is made up of molecules called nucleotides. Each nucleotide contains a nitrogenous base, a phosphate group, and a pentose sugar. DNA contains 4 different nitrogenous bases which contain atoms of carbon, hydrogen, oxygen, and nitrogen. These 4 nitrogenous bases are adenine, thymine, cytosine, and guanine. Adenine and guanine are larger double-ring bases called purines, whereas thymine and cytosine are smaller single-ring bases called pyrimidines. The nucleotides are named according to the base that is present. For instance, a nucleotide containing thymine is called a thymine nucleotide. One containing adenine is called an adenine nucleotide, and so on. The order of these bases is what determines DNA's instructions or genetic code. When we talk about pentose sugar, a 5-carbon sugar called deoxyribose attaches to each base in DNA. Phosphate groups alternate with pentose sugars to form the backbone of a DNA strand. The bases project inward from the backbone chain. DNA and RNA store genetic information as sets of 3 nucleotides. Now, what are the functions of DNA? There are 2 important functions of DNA. The first one is that DNA serves as a genetic blueprint during cell replication. Through this role, DNA ensures that the cell produces cells just like itself, thus continuing the identical type of cell line within the body. Furthermore, in the reproductive cells, that is eggs and sperm, the DNA blueprint passes on genetic characteristics to future generations. Next function is directing synthesis of protein. DNA provides codes or instructions for directing the synthesis of specific structural and enzymatic proteins within the cell.